So basically, as you know, you have three parts. One is Dr. Shahida and Dr. Yusri for the statistics, and my part will be the biological part of the forensic subject. Basically, we're going to try to use DNA to find out things, But, there's no notes. So we're going to have theory and labs. I think we're going to have 3 labs. So like I said before, I already changed everything, the structure of the lab and the lecture. We are going to do lecture first, and then once the lecture is done, then we can do lab. So the good thing about this thing is that everything is almost standard. DNA stuff is almost standard. So we already have all the links. Like a lot of people has done YouTube courses on this. So I put this link for you guys to watch it later. Don't worry about this. This looks complicated. It's very simple. And these are basically the topics that you're going to learn. Genes, DNA isolation, polymerase chain reaction, sequencing, blast, this one. Don't worry about this. It's all very simple. And this one, and this one. And hey. Okay, but this is what I'm planning to do. Yeah. So before we start, I think it's very important to understand all these concepts, because I'm sure you have learned this at your A level or maybe before, and about cell, nucleus, chromosome, genome, gene, DNA, nucleotide, base, GATC, ribosome, sugar, phosphate, RNA, transcription, expression, ribosome, translation, amino acid, protein. Have you heard about this all? Yes. Do you know what they are? Do you clear? Okay, I'm going to ask the simple questions. Does anybody know the difference between DNA and RNA?
Yes, the shape.
The what? The shape. Anything else? Okay. The sugar. What about the purpose, the functions? Do you know the difference? Replications, okay. What else? What? To make protein. How do you make protein? Okay. What about gene and DNA? Do you know the difference? Are you clear? Because it has to be cleared, before we can learn this. Or chromosome or genome. What are those?
Yes, the function.
Okay, what's the function?
Carry blood. DNA is to
Carry blood. No. Okay. Okay, I'm going to explain. This is basically all that we're going to learn. This is without understanding this, you'll never know what DNA and what the stuff are. So basically, this is your cell. Okay, so this is the repetitions of what you've learned before. But I will try to put them all together. This is your cell, and inside your cell you have cytoplasma and nucleus. Okay? And this nucleus, it contains all your genetic markers. Okay? And then all the genetic markers are in the forms of, what is this? Chromosomes. Yeah. And then chromosomes, if you extend them, you extend all your chromosomes, it will become? This is your genome. Basically the genome that contains all your genetic markers. Like basically if you extend them, I think there are like billions of sequence. You don't have to write this. I just want you to understand the concept first. Because without this, it's going to be difficult. And genome is made of? Genes, yeah. So what's the difference between a gene and a genome? Single and plural. Okay, so basically the building block of your genome is genes. So if you put genes together, one after another, and once it's complete, that's your genome. So basically, genome contains your whole genetic marker. And genome is a fragment of your, not genome, genes. Genes is basically a fragment of your genome, okay? Is that clear? Okay, but what's a gene? How I mean, is it only a fragment? No. Okay, what else is important? Carry? Identity, of course, carry what? Sorry? Okay, so basically, genes, the building block comes in a specific function. So you have genes for making eyes, you have genes for making feed, you have genes for making you fat, genes for making you male and female and male, yeah, all those, yeah. So basically, genes is a fragment of DNA that comes with specific function. So with genes, you have function. So basically, this function or this information is in your genome, and you already know what you're capable of in the forms of a gene. So do you know now the difference between a gene and a genome? Is that clear? So a genome is a complete sequence, and genome comes with a specific function. But these are your master copy. Basically, these are your genetic markers. So what's the difference between RNA and DNA? Besides single-stranded and double-stranded, what else? And of course, the base is different. What about in terms of function? Okay, if you want to make a, how do you say? If you want to make a cake, you have a mold, okay? And you want to make a cake. Will you use that one mold that you have, which is your master copy, or will you copy the master copy and use the second copy to make the cake? Because if you keep on using your master copy, you're going to lose it. So you need to produce a new, not really master copy, the second copy, and then use it. Does that make sense? That's what exactly what our body is doing. Because if you use, I mean, I'm just trying to make it simple. If you use the genes, the DNA, to produce the protein, not really, because you only want certain parts of it. But it's okay. I would say like, so this is your master copy, but then before you make a protein, you're not going to use your master copy, but you're going to replicate or duplicate your master copy and send this master copy to the factory and produce the protein. So that is when you basically replicate the DNA into RNA. They call it transcribe the DNA into RNA, and then you will use this RNA as a mold and send it to ribosome, which is the factory, and then do a process called translation and make the protein. So do you know what's RNA now? Okay, do you know what's a genome? It's actually very important to put all the things together, you know, before we can start details. We're going to watch the video after this, so don't worry if you're not. All right. So, why can we use DNA in forensic? It's actually very important. How can we use genes in forensic? Why? Different people has different genes or different genetic informations. Do you all have, does all of us has different genes? Is it 100% different? No. Okay, that's good actually. How many percent do you think is different and how many of them are similar? 70. Actually more. How many eyes do you have? 2. So how many eyes does she has? 2. So the genes that control the number of eyes, is it going to be the same or not? Yes. How about nose? Mouth? Okay, so basically our genes are very similar. But then, so basically I would say like if you have this complete genome, maybe 99.9 is the same. And then, but there are some differences, you know, like your fingerprint. Is your fingerprint the same or different from your friends? No, okay. So out of all the genes, for instance, like this is controlling the number of eyes, controlling the number of legs, controlling the color of your hair. So it might be the same, but then you have this one at the end that control the pattern of your fingerprint. So is this going to be the same? No. So if you want to compare between two people, are you going to compare these genes that make eyes? Oh, okay. So you're going to compare the one that differentiate your fingerprint. Okay. So that is called hypervariable region. Yeah. Hypervariable. That means like it varies a lot. So to differentiate between two people or two bacteria or two species, you only compare this. Yes, the hypervariable region. So there is no point comparing the whole because it's going to be the same. It's redundant. Okay? Does it make sense? Okay, so if I tell you one thing, I said like because we have big issues about halal food. Okay, if I tell you that in our canteen in Bhakti, there is a pork gene or pig genes. Is that true or not true? No, because I can write in a newspaper saying that I found pig genes in the canteen at our Bhakti canteen. And it's actually true. It is true. Why? I can just write like that, you know, I found pork genes in all canteens of USM. And I'm not lying. Because, look at this.
Probably like 70% similarity between us.
Exactly. So there are similarities between pork gene and human. Maybe, I don't know how many percent, maybe 80% or maybe 90%. So the genes that the pork has is the same as human or the same as chicken. But if I say that I found genes specific to pork in the canteen, then that can be a problem because it's only specific to pig. So be careful. All right? So in our whole course, basically, what we're going to do in the lab, we're going to teach you on how, after understanding, this is basically all you need to know. So we're going to teach you how to extract DNA and compare this hypervariable region between different species, or because some genes are controlling of production of certain things, you know, like certain protein or certain toxins, and you can check whether they have the genes and see their capability. Basically, that's all you need to know for this lecture. And how do you extract DNA? Okay, so basically, you're going to learn DNA extraction in the lab. So basically, after this is your theory, and then we're going to teach you the lab. DNA extraction is very simple. Like Heidi said, lyse the cells. So basically, DNA is easy to extract because of only one property of DNA, which is very, very important, that makes extraction very easy, because of alcohol. Actually, DNA in alcohol, it's going to precipitate or clump together and become big. That's in alcohol. But in water, they're going to basically dissolve in water, and they become small. So this characteristics is very important in DNA extractions. So like Heidi said, first you need to lyse the cells. If you have cells, because these cells contain, you know, any cells contain DNA, and then you basically need to lyse them, so to break them. So if you have like these cells, this one, you want to take this one. So you need, first of all, you need to break the cells. So you can do many ways, like you can use enzyme, you can use soap, you can use liquid nitrogen, you can crush them, you can destroy them, you can heat them up, basically to break all the cells. And you're going to have a mixture of all the cells with DNA combined together. But then you need to split the DNA and with all the rubbish, I would say, like the rubbish that you have. How do you do that? Remember the property in alcohol change, you know. With alcohol, it's going to be big. So if you have like this one, like your DNA, and then you have all the other rubbish, you're going to have big rubbish, and you're going to have small rubbish. Okay, so this is your DNA. So if you put alcohol, the DNA is going to be like this. So if you put them into, if you have like a sieve, says the diameter is bigger than the alcohol, than the DNA, if you sieve them, this one will go through. All right? And then this DNA and the big rubbish will stuck. All right? Up there. And then if you remove the supernatant, and then you have a mixture here of your DNA plus the big rubbish. Okay? But remember, with alcohol, the size of the DNA is big. But then if you put water, these big ones, it's going to become small. But this one doesn't change. So if you put water, so the DNA will become small and go down, you can collect it, and the big one will stay here. That's basically the principle of DNA extractions. Alcohol is so important because it makes the size bigger. they call it, I just try to make it as simple as possible. Of course, like, it's not that simple, but that's the basic principle. You need to know, because in the lab, you need to know when to use or not to use the alcohol. Okay, so once you get this DNA, you can find the genes, but we can have the DNA, and we can find the gene sequence, but it's okay. The aim of this part is to let you know that having no basics of, not very good basics of biology, DNA work is actually not that difficult. It's actually, basically, this is all you need to know, and this is all you need to know. It's actually quite simple. Let's see. So are you clear now what this one is? Cells? Okay. Nucleus? Already know. Chromosomes? You know. Genome? Okay. Genes? DNA? Oh, nucleotide. Did we mention? We haven't talked. Okay. So nucleotide, you have these genes. What's the building block of these genes? Sorry? Yes. What is a GATC? It's not DNA. It's making DNA. Also, actually, nucleotide is the building block of the DNA. Like, because you have genes, you have sequence, like this long, and then the nucleotide is a single building block of it. And the nucleotide is made of one base, one ribose, and one sugar phosphate. But this is detail, yeah, which you're going to learn, and yeah. And you have 4 different types of base. And you know RNA now? Do you know? Okay, so you basically transcribe, you replicate the DNA into RNA, and then you use that to make protein synthesis, yeah. And, okay. Oh, this is important, actually, gene expressions. Okay, this process of making RNA from DNA is called expressions. This is important because having the gene itself, does it mean that you will always produce the protein? Like, if you have a gene for gray hair, like mine, but I colored it. Okay, will you produce gray hair all the time? No. Okay, so there are things that you have in your genes, but you don't produce it. Does it make sense? You have maybe genes for cancer, but you don't have cancer. Or you have genes for getting fat, but you're not fat. So you need something for your brain to decide what to produce. And the production of that RNA from DNA is called transcription, and I would say it's gene expression, So gene expression is the time when the protein is, the RNA is actually being expressed. And when your body decided to make the protein. So it's going to first transcribe the gene into RNA and send it to the protein. And when you, in the ribosome, when you make the protein in ribosome, that is called translation. And you make amino acid, and which is the building block of protein. And if you combine a lot of amino acids, it will become protein. That's how you make your protein. And remember, like every sequence, like G, A, T, C, 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 that will produce one type of protein. Like, so certain sequence encode for certain protein. So that's why when you have the genes, but your body, the sequence in your genes, so basically certain codes will encode for certain protein. So that's why the sequence from the very beginning is very important, because that decide what protein is going to be produced. We're going to extract DNA and find these genes and then compare it with the database to see what they are. We're going to do that in our lab, actually, yeah. It's actually very good. I'm glad you're a bit advanced. We're going to do that, yeah. We're going to do that. But for now, I want you all to understand the basic concept.